Guys, now that you have learned that how citrate is formed in when some of the very initial reactions of the TCA, now let's move a little bit ahead. Once the citrate is formed, it undergoes isomerization. Isomerization means the molecular formula changes, but the structural formula changes, but the molecular formula remains the same. And the enzyme is the aconitase. Aconitase is an iron sulfur protein which isomerizes the citrate to isocitrate. Also keep in mind that fluoroacetate is a plant toxin which is used as a pesticide here. There are many pesticides or insecticides which target the different enzymes of the Krebs cycle, because a Krebs cycle is so important in respiration of organisms. So you can really target those enzymes if you really want to get rid of those organisms, you want to kill them, so that you can really have a better crop or whatever you want. Then the second reaction, after the formation of the isocitrate, is a very important reaction, by the way, oxidative decarboxylation of isocitrate. Here isocitrate is converted into alpha ketoglutarate in the presence of enzyme isocitrate dehydrogenase. A carbon molecule is released, and in addition to the carbon molecule, you also get the NADH2. So this is the first reaction of the Krebs cycle or of TCA where NADH2 is formed, and you know the importance of NADH2. It's a very important regulatory step. It is allosterically activated by the ADP and calcium ions, and it is inhibited by ATP and NADH. So you can envisage that why ATP will acting as a inhibitory molecule here. Then in the next reaction, alpha ketoglutarate is converted into succinyl CoA, and the enzyme here is alpha ketoglutarate dehydrogenase complex. It's almost the same sort of structural aspects this enzyme have as the other dehydrogenase complexes, like pyruvate dehydrogenase complex. It is inhibited by NADH and succinyl CoA. Of course, these are the products. Again, you should remember what I said about the negative feedback mechanism, and activated by the calcium ions. By the way, the last reaction was the second reaction in which NADH2 was produced, and again you know what is the importance of NADH. Then the cleavage of the succinyl CoA takes place under the action of the enzyme, what we say, the succinate thiokinase. In this reaction, GTP is formed. This is known as the substrate level phosphorylation. Importance of GTP is that it can readily be converted into the ATP, which again is one of the reason that metabolic pathway runs, because we need energy.